This is the last lecture for the segment where we're looking at genetics, we're looking at inheritance of genetics, passing on from one generation to the next and the like. After this lecture, the rest of the semester, the last few weeks that we have, will be primarily focused on evolution and ecology. So this one's going to take a little while though. As such too, your quiz is a lot longer than other quizzes because there's some problem solving that you're going to have to do in this one, namely working with Punnett squares and the like. So let's review our definitions on some of these concepts so that we can understand how it is that these how we can predict with a high degree of certainty the outcome of when you have two individuals. Even though we pass on genetics randomly, we can still make predictions on the probabilities of what things are going to be passed on because they follow certain laws. So there's laws of inheritance that were discovered actually 200 years ago that still hold true today, even before they understood what DNA was and what genes were and whatnot. So I'll give you a little background history and a little bit of that. For now, let's just review. So chromosomes, remember, are in eukaryotic organisms are these discrete segments of DNA that are wrapped around proteins. We have 46 of them. Other organisms have more or less. Again, it doesn't come down to how many you have. It comes down to how much of those is actually composed of genes and how those genes are transcribed and translated and spliced and all that kind of stuff we've talked about. So chromosomes have segments within them that are all the information necessary for the cell to be able to make a protein. So remember that a gene is that sequence of DNA, those nucleotides that encode a polypeptide chain. Now, remember also that only 2% of our genome actually encodes genes. So of the 46 chromosomes, only 2% of it is actually made of these segments that encode proteins that the cell uses to, do everything that it does. Now, we brought up this term last time, and this is what's going to prevail in this lecture as far as understanding inheritance is alleles. Now, we've talked about mutations and how when you get something like a point mutation or a frameshift mutation, that changes the sequence of the nucleotides by adding one, removing one, substituting one. Ultimately, that change in the sequence of nucleotides can change the overall protein and its function and its role. A lot of times it just kind of messes it up, but there are other times where it doesn't and you get some type of new function. They're few and far between when that occurs, but that's occurred over a long period of time. We call these alleles. So if you look at chromosomes, like these two, and you look at specific locations on homologous pairs, you'll find that you have the same type of gene in the exact same locations on the homologous chromosomes. Okay, and that's really the definition of homologous chromosomes. They're chromosomes with the same size, same shape, same amount of DNA, and the same types of genes. So if you have a gene for hair color here, you've got a gene for hair color there. If you've got a gene for eye color there, you've got a gene for eye color there. Okay, so these are very specific proteins, but due to the fact that you've inherited these from separate parents and the nucleic acid sequence might have mutated somewhere in their past, the alleles might be different. Now, they don't always have to be different. Sometimes two parents pass on the same allele, but that's the nature of alleles. Alleles is what version of that gene do you have. And you're going to see several different examples of that today when we look at traits that are being inherited, such as blood type, and hair color and things of that sort. Okay? Now, let me forewarn you, a lot of times people will ask questions about their parents or their friends' parents that might reveal things that you might not want revealed. So make sure you know what you're asking so that you know what the answer is going to be. Because we've had people ask, well, both of my parents are blood type O and I'm blood type A. And then it gets awkward because there are circumstances. So that's a possibility that you are the biological offspring, and we'll show why, but just be aware of that. All right. Now, alleles. This is what we're primarily going to talk about in inheritance. Not all alleles, now since we have homologous chromosomes, which means we have 2 of every gene, we have 2 alleles. Now the alleles might be the same, the alleles might be different. It all depends upon what your mother and father passed on to you, okay? Now, the effect that these alleles have on one another comes down to certain terminology that we use, such as dominant and recessive. So, every person has 2 of every gene. You can see that here from the homologous chromosomes, is you get 1 allele from your mother and 1 allele from your father, okay? If the alleles exert a dominant influence, then we call it a dominant allele. So what is a dominant allele? Well, a dominant allele is one where it is expressed no matter what. Okay, so it's expressed, let's say like brown hair is a dominant trait because it's a dominant allele. Whereas things like, let's say, eye color, blue eyes, is a recessive trait. Brown eyes is a dominant trait. So the alleles that make your eyes brown are dominant. So if you have 1 allele for brown eyes, and now I'm simplifying eye color. It's not down to just 1 gene, but I'm simplifying it for the sake of examples. If you have 1 allele which is for brown eyes and 1 allele for blue eyes, your eyes are going to take on the dominant allele's expression, in which case you would have brown eyes, which is why we say brown eyes is a dominant trait and blue eyes is a recessive trait. Now we know that there are many different types of eye color, and I can talk a little bit later more about how that's done, but for now we'll keep it simple. Now recessive traits can only be manifest if the person has 2 recessive alleles. Because if even one of them is a dominant allele, then it supersedes the recessive allele. So the recessive allele always gets masked in the presence of a dominant allele. Okay? Now, here's one thing you have to understand. Recessive doesn't mean least frequent. In fact, some recessive traits like blood type O and blue eyes are actually more common than the dominant traits. So in Europe, blue eyes is the most common, even though it's a recessive trait. Here in the US, blood type O is the most common blood type, even though it's a recessive trait. So recessive doesn't mean infrequent. It just means that if the only way to get that trait is if both of your alleles are recessive. Now that brings us up to a way of describing the makeup of our alleles. Since we have 2 of each, we have terminology which ultimately defines the difference between what we call genotype and phenotype. If you think of genotype, geno as the genes, the genotype is essentially asking the question, what are your 2 alleles? Which ones did you get from your parents? Now, if your alleles are the same, you got the same allele from each parent, we call that homozygous. However, since there are 2 different types of alleles, dominant types and recessive types, you have to quantify what type of homozygous it is. So if it's homozygous dominant, that means that you have 2 dominant alleles, and that's homozygous dominant. If you're homozygous recessive, it means your alleles are recessive, and you have 2 recessive alleles. So you have to say whether you're homozygous dominant or recessive. You can't just say, oh, I'm homozygous, because that can mean 2 different things. Now, heterozygous, that only means one thing. It means the alleles are different. Now, you might think, well, why don't you say one allele is dominant, one's recessive? You'll see later on, it's not as simple as everything being dominant or recessive. So it's easier just to define it now as saying heterozygous means the alleles are different. Okay? And that's it. You could just say, oh, I'm heterozygous. That means that your alleles are different. Okay? Now, the phenotype. This is what's being expressed. Now, even though it says outward expression, that doesn't mean all of your traits are on the outside. For example, your blood type. That is not an outside look, That's what's going on inside your body. But your blood type is a phenotype, okay? And we'll talk about blood type today. So, phenotype just means what is the manifestation of the combination of your 2 alleles. If you're homozygous dominant, then you'll typically have a dominant phenotype. If you're homozygous recessive, then you'll have a recessive phenotype. And if you're heterozygous, well, if one of them's dominant, one's recessive, then you'll have a dominant phenotype. So the phenotype is essentially, when you put these two alleles together, what's the outcome? All right, so here's an example. This works for plants, this works for people, and whatnot. We're going to talk a little bit about both today because a lot of the experiments that were done to figure out how inheritance works were done on plants first, and then we apply that to people as well. Now, let's say that there's a gene that ultimately can make you determines your height. Now, one version of the gene makes you tall, the other version of the gene makes you short. Well, the tall version is a dominant allele. But notice, the genotype for these two individuals who have the tall phenotype are different. This person is homozygous dominant, this person is heterozygous. But here's the thing, you wouldn't be able to tell by looking at them which person was which. So having two dominant alleles versus one dominant allele doesn't make the person doubly tall. They both have the same phenotype. So make sure you understand that two dominant alleles versus one dominant allele, the phenotype will be the same. There will be no difference between two individuals. And the same thing is true when we look at things like blood type. You could see two people with blood type A, but you wouldn't know whether one person's homozygous or another person's heterozygous. You couldn't tell because their blood type is exactly the same. Over here, the short allele only shows up, or the short phenotype only shows up when both of the alleles are recessive. Because if you have a recessive allele, but the other one's dominant, you're going to have the dominant phenotype. Okay? So make sure you know this terminology. It's going to come up in most of your scenarios where we talk about the person's genotype or their phenotype. So again, genotype is what are the alleles. Phenotype is what combination do those alleles cause to happen. What's your blood type? Are you tall? Do you have brown hair, blue eyes, whatever the case may be? That's the phenotype. Now, a little background history before we get into the laws. These were discovered centuries ago by a monk who had a lot of time on his hand, gardening and whatnot. And he would breed plants over and over and over and actually take statistical data. He was the one that actually coined the phrase alleles. This is before they understood what DNA was, that there even was something like DNA, or even something like genes. So that's why he called them alleles. Now, we still call them alleles today, but we understand them in more modern vernacular than he understood it back in the day. But the monk was Gregor Mendel. He's really kind of the father of inheritance and genetics and whatnot because of all these experiments that he did. And in fact, a lot of this information was lost for a while. Someone else did these experiments later on, discovered it, but then they found his experiments and were like, oh, well, he gets the kudos because he did it so long ago. Now, a lot of his experiments were done with the pea plants, or that he cross-pollinated and bred with one another. Now, before I show you a couple of the experiments that he did, let's talk about the laws that he came up with. There are two. Now, remember in our progression of the scientific method, these laws ultimately give us such a good understanding of that concept that we can make predictions. And that's really what inheritance is all about. How is it when you go into the doctor and he sees your genetics and he says, oh, you have a 1 in 4 chance of having a child with cystic fibrosis? How can he make that prediction? Well, it's based upon these laws of inheritance that are universal for any organism that undergoes sexual reproduction. So let's talk about the first law, the law of segregation. This is where we're going to spend most of our time this lecture in terms of application. The second law is important, but we're not really going to deal too much with it in terms of making predictions and whatnot. So, law of segregation. You've actually learned this from the last chapter when we talked about meiosis and how alleles ultimately get separated from one another during the meiotic process where homologous chromosomes are separated during anaphase 1. So that's really all the law of segregation is that we have two alleles, but every time we undergo sexual reproduction, we only pass on one of those two, every time. Okay? So you may start off, you may be heterozygous, And every time you have an offspring, it's essentially a 50-50 chance of which one you pass on. You can either pass on that one, or you can either pass on that one, but you don't pass on both. Okay, so that's the law of segregation. Why is this applied to meiosis? Because each of these alleles are on a different homologous chromosome. And during anaphase 1 in meiosis, when homologous chromosomes separate, the respective alleles also separate, and you don't inherit two from the same parent. So that's the law of segregation, that ultimately each parent only passes on one allele. So that's manifested here. Now, obviously if the parent is homozygous, the law still applies. They only pass on one of their dominant alleles, but as far as statistics go, it won't make any difference. They're going to pass on the dominant allele either way. And same thing is true if someone's homozygous recessive. They'll pass on the recessive allele no matter what. So statistically, that's all they can pass on. It's when you have a heterozygous parent that it really comes down to a 50-50 chance. Half of the gametes are going to have the dominant allele, half of the gametes are going to have the recessive allele. When you combine these two together, then you have certain outcomes that result from the fertilization process. That is the law of segregation. Now, there's a way in which we apply this, and this is what about 7 out of your 10 questions are going to be like, is the Punnett square. Okay? The Punnett square, let me make sure you write this down because this is key. The Punnett square is the same as the law of segregation. Okay? The Punnett square is the application of the law of segregation. Okay? So what you're seeing here is the genotype of the two parents. Well, this is how we calculate the statistics, is we split up the two alleles from each parent, and then we combine them in this square, called the Punnett square, and that gives us the probabilities of the outcomes that result from that. So, here's what Gregor Mendel did. He took some pea plants and he was looking at various phenotypes, like what color were the pea pods, or the peas in the pod, or how tall were the peas, or were they round versus wrinkled or whatnot. And they were very quantifiable phenotypes. Well, he found that some plants were what he called true breeding plants for certain traits, meaning every time he crossed two plants that had yellow peas, they always had yellow peas. And every time he crossed certain green pea plants that had green pea pods, they always were green. Well, the reason for that is because each of those parent plants were either homozygous dominant or homozygous recessive. For example, if you cross two yellow plants, or two pea plants that have yellow seeds, there's no other outcome but yellow seeds. That's how he initially saw that these were true breeding. Same thing true for the lowercase y, which is why we denote recessive, is that when you cross green plants with one another, or plants that produce green pea pods, the only outcome was green peas. But what happened when he crossed that true breeding yellow with the true breeding green? Well, the end result was he still only got yellow seeds. However, there was something different about them. Oops. I want this to be little y. And this to be big Y. When he crossed these plants that only produce yellow peas versus green peas, they were all still yellow. Now, why is that? Why do you think, from what you can see here?
Yellow was the dominant trait.
Good. Yellow was the dominant allele, and no matter what, you were always going to have a dominant allele with the recessive allele. Now, this is the second generation. This is what he discovered. When he took the offspring of those plants and bred them together, all of a sudden the green peas started showing up again. And this is the reason why. He called them alleles, and he knew that they were being passed on from the parents. Again, now we understand them as variations of the same type of gene. But here's why. So here we have a heterozygous parent. Here's the other heterozygous alleles. And when you put them together in the Punnett square, you have a 25% chance of being homozygous dominant, 50% chance of being heterozygous, and 25% chance of being homozygous recessive. And that's what he saw in his outcomes, is every time he would cross-pollinate these and he would count the pea pods, and it was a lot of peas, thousands upon thousands, he got this 3-to-1 ratio. He got 75% of the seeds being yellow and 25% of the seeds being green. Now notice, when he counted them, he was counting the phenotype. Notice too that the genotypes here are different between these, yet their phenotype is exactly the same. And that's what we've talked about with the dominant allele. Doesn't matter whether it's 2 dominant alleles or 1 dominant allele, the phenotype is exactly the same. So here's how you do a Punnett square. When you have an offspring from 2 parents, you essentially put the alleles on the 2 sides of the square. Now it doesn't matter male alleles here, male alleles there, female alleles, doesn't matter, but just choose a side. Okay? So in this situation, here we have the male probabilities. Because he's heterozygous, he's going to have a big Y and a little y. And because the female is also heterozygous, she's going to have a big Y and a little y. So here's how you put them together. You just take this big Y and put it down this column, this little y and put it down that column. Okay? This little y, put it down that column. This big Y will go down this column, and this little y will go down this column. Now, for the sake of simplicity, we usually put the dominant allele first, but you get the same outcome. It doesn't matter whether you write it like this or whether you write it like that. It's the same genotype. Okay, so don't think that these 2 are different genotypes, which is why you'll typically see me do something like this. Even when I'm putting these across, you'll see me put it off to the side because I know that a dominant allele is going to come in there. It just makes it easier to read. So here's where we get probabilities. 1 in 4 chance of being homozygous dominant, which is equal to 25%. This is where your math comes into play. It's really simple, but 2 out of 4 chance, which is 50%. Now on the test, I'm not going to do 1 out of 4 or 2 out of 4. I'm going to do percentages. So it'll be 25%, 50%, 25%, and whatnot. Okay? So this is what Gregor Mendel did with these true breeding plants. And then when he crossed them again, that was the previous slide. Here is the one here's another example of a heterozygous parent with a homozygous recessive parent, and you end up getting 50-50. You end up getting half of the seeds being green and half of the seeds being yellow. Okay, so there's just some different things. We're going to do a lot of Punnett squares as we go through here. So now, we're not going to do any Punnett squares with the law of independent assortment, because in reality, the law of independent assortment doesn't really make predictions, but it does talk about one of the natures of inheritance that must be considered. Okay, so you've seen that word before, independent assortment. Where have you seen it? Or I told you about it before. You should know it for this week's quiz.
Independent, this is your probabilities.
Yeah. You're getting close, but not quite. What happens during metaphase 1? What line up along the middle? Homologous chromosomes line up along the middle. Remember we talked about this as a recombination event, independent assortment. It's one of your 3 recombination events: crossing over, independent assortment, and fertilization. So why is this a law? What is this law about? Well, the law essentially states the following thing. The inheritance of one gene does not influence the inheritance of another gene. If I look at you and say, oh, you got brown hair, so you're most likely blood type A. No, they have nothing to do with each other, because due to the fact that chromosomes can line up however they want along this middle, that's the law of independent assortment. That genes do not influence the inheritance of other genes in their when they're being passed on. So I can't look at you and know what your blood type is based upon your hair color or based upon your height or anything else of that matter. There's no correlation between the two. Now, we can show this by doing a really difficult Punnett square, but you're not going to be doing things like this. So I found that it's just simpler to just show you the complexity of it and then pass it by. So, but I do want to illustrate this process really quickly. Remember, this happens during metaphase 1. So here's an example of the P's that Gregor Mendel did. And this is what he found when he was doing these experiments, is he was looking at lots of different traits. And when he looked at the seeds, he found that some were yellow, some were green. He also found that there was another trait. The seeds were either round or they were wrinkled. Okay? So the yellow, remember the yellow or green, we denominate with the Y gene. The yellow being the dominant, the green being the recessive. Over here, we've got the wrinkled or round. Now, we found that round was a dominant, so the big R symbolized the dominant, and wrinkled was recessive, and so the little r represents the recessive. Well, here's one scenario. If the homologous pair line up like this, and then the homologous chromosomes separate from one another, then the outcome is this. The seeds that inherit these alleles will have the yellow round, and the seeds that inherit these alleles will have the green wrinkled. But that's not the only way that they can recombine. Here's the alternative, where these flip. Now, the seeds that inherit these alleles will be round and green, and the ones that inherit these alleles will be wrinkled and yellow. So you can see that the color of the seeds doesn't influence whether or not they're going to be round or wrinkled. The two genes are assorted independently of each other. So that's the law of independent assortment. And so he showed that by doing these more complex scenarios, but you would be able to find, if you analyze this, that there was no difference in the inheritance of one or the other. Okay? Now, there is an exception to the law of the law of independent assortment. And that is if you're looking at two traits that are influenced by two genes and the genes are on the same chromosome. That's the only exception to this law. Why? Because it doesn't matter then where they flip in relation to each other. Because the genes are on the same chromosome, they'll flip together because they're on the same chromosome. So the inheritance of genes that are on the same chromosome is the exception to this rule. Now, we're not going to do any exceptions to these rules, but there is a way of recombining these alleles still. Do you know what? Do you know how? Crossing over. You could still get crossing over, and that was still mix and match. Now, it's not as probable, but it does happen. And so you do get recombination. And in fact, they've shown where the genes are on the chromosomes by the percentage and probability of crossing over between them. And they find that the more they're inherited together, the closer they are on the chromosomes. This is before we were able to sequence all the DNA and know exactly where they're at. Now we know exactly where they're at. All right. So, most of the time, when people do the law of segregation, where they're making predictions about the outcome of their child, they don't care, they're not worried about what hair color the child will be. They're not worried about what their eye color will be, or typically not what their blood type is. They're more worried about if the parents' genetics recombine in such a way where they'll inherit a genetic problem, a genetic disorder, like Huntington's disease, and sickle cell anemia, and cystic fibrosis, and Tay-Sachs. These are the concerns. So when we look at the family pedigrees, this is where genetic counseling comes into play. Because if you know you have Huntington's disease, and you go and get tested, and they say, oh yeah, you're heterozygous, Huntington's disease is a dominant allele, which means that if you're heterozygous, you have a 50-50 chance of passing that on to your child. And this is where genetic counseling comes into play. And this is true for any type of genetic disorder, where people are informed of the statistical probability of having a child. They may look at both parents' genetics and say, you have a 75% chance of this child having this, and then allow them to make that decision on whether or not they want to have any children. So let's look at some genetic disorders. Most genetic disorders are not this simple. but we're going to use the simple ones. Okay, so there's a lot of ones that are caused primarily by a single gene, but not all genetic disorders are caused by a single gene. For example, you may say, oh, what are the chances that I'm going to have a heart attack? Well, there are many things that lead to a heart attack. Okay, it's much easier to say, what are the chances that I will have something like sickle cell anemia? That's easier because it's down to one gene. What are the chances that I'll have Huntington's disease? That's easy because it's down to one gene. So most of the genetic disorders that we use this for are very quantifiable, very predictable because they're single gene mutations. They're very studied, they're very well studied. But there are many others that we can make predictions, but they're more complex than for this class than we need to do. Now, just like there are traits which are dominant and traits which are recessive, diseases also follow the same rules. Some diseases are dominant and other diseases are recessive. Now, we're also going to show that there's a difference on which genes are inherited. Some mutations, because most of our chromosomes are non-sex chromosomes, these are the ones that we look at quite often, are what we call autosomal disorders. Remember, autosomal chromosomes are the non-sex chromosomes. But we're going to look at both. We're going to look at disorders that are found on the autosomal chromosomes and disorders found on the sex chromosomes. When we say a disorder on the sex chromosomes, we usually call it sex-linked disorders. So autosomal disorders, doesn't matter what the sex of the individual is. Sex-linked disorders does. It does matter whether you're male or female because it influences us differently depending upon what we inherit, and you'll see why. Now, you're not going to have to memorize any of these nor explain what the outcome is, but here are several quantifiable examples of genetic disorders that we look at. Let me just go through a couple of them. Achondroplasia. This is actually a dominant disorder that causes dwarfism. In fact, 95% of the time, dwarfism is caused by this mutation, by this problem right here, achondroplasia. There's Huntington's disease. That's also a dominant disorder. Marfan syndrome. This one actually has many, many, many different symptoms, most of them being abnormalities in the skeletal system, but there can be other abnormalities as well, depending upon some of the dynamics. Polydactyly, extra digits in your fingers or your toes. Now, these are dominant disorders. So let's look at a scenario that you may have. Let's look at Huntington's disease. Let's see why this is so problematic. Now, one of the reasons why Huntington's disease has not been weeded out of the human genome is because it doesn't show up until after reproductive success. So it's not something that prevents you from having children. It's something that basically affects you later on in your life after you've passed the genes on. And that's why a lot of times people, if it runs in the family, will get tested so that they know whether or not they want to have children and have that probability of passing it on. All right, so in this scenario, the normal phenotype is recessive. To have a normal brain function and not any problems with this, then it's recessive to the actual problematic allele, which causes Huntington's disease. Okay, so Huntington's disease can be caused by either a homozygous dominant or a heterozygous. Most of the time it's heterozygous. Most of the time. But that doesn't preclude the possibility that somebody's homozygous for this dominant allele. Again, it doesn't give someone a higher probability of having Huntington's disease by having two dominant alleles versus one dominant allele. They'll have Huntington's disease one way or the other. It doesn't, you don't get it sooner, you don't get it more severe. It doesn't affect whether or not you have it. Okay? So, you're going to see throughout a lot of these questions too, when I say that someone has a normal phenotype, because I've been called out on this before, they're like, what's really normal? Normal means you don't have the disease, okay? So, if I say normal phenotype, it means they don't have Huntington's disease, they don't have polydactyly, they don't have cystic fibrosis, they don't have sickle cell anemia, okay? So, we'll make that clear. Now, let's do a scenario. Let's say that let's start very simple. Let's say neither parent have Huntington's disease. What is the probability that they'll pass it on? If neither parent have Huntington's disease, what's the probability that their children will have it? 0. Okay? So you can see that people that don't have it, don't have a history of it or whatnot, it doesn't just all of a sudden pop up. Okay? It doesn't show up. All right. Now, let's say one parent has Huntington's disease and they are heterozygous. That's a qualifier that I have to give. Why? What if I say a parent has the phenotype Huntington's disease, but I don't tell you anything more? Why is that a problem? Why do I have to tell you what their genotype is? Because it's a dominant disorder. What's the scenario? Either one of these genotypes causes Huntington's disease. So if I say, a parent has Huntington's disease and leave it at that, you don't know which one it is. Okay? So that's the scenario with dominant disorders. You have to say, and they're homozygous dominant. And they're heterozygous. So you'll see that in some of these questions where I'll qualify, their genotype is heterozygous. Okay, so let's do that. One parent has Huntington's disease and is heterozygous. The other parent does not have Huntington's disease. They have the normal phenotype. Okay? So, the parent with Huntington's disease who's heterozygous, what are their alleles? Big H. Now, they're heterozygous. Good. Little h. Remember, the alleles are different. The parent who's normal doesn't have Huntington's disease. What are their alleles? Good. Little h, because if even they have one dominant allele, they're going to have Huntington's disease. Okay, big H down this column, little h down this column, little h across that column, little h across that column. What are the chances that they'll have a child with Huntington's disease? Good. Okay, now let's say both parents have Huntington's disease and both parents are heterozygous. What are the chances that they'll have a child that's normal, no Huntington's disease? So let's look at their scenarios. Both parents have Huntington's disease and both parents are heterozygous. What are their alleles? Big H, little h, and big H, little h. Okay? Big H across there, little h across there, big H down that column, little h down that column. What are the chances they'll have a child with a normal phenotype? These are the statistics that parents want to know. These are the things that genetic counselors essentially show them. You have a 25% chance of the child not having Huntington's disease. Okay? All right, so those are dominant disorders. Whether it's Huntington's, whether it's achondroplasia, whether it's polydactyly, the principle is the same. One dominant allele causes the dominant disorder. Now, that's not the situation for autosomal recessive disorders. Like recessive traits, you need 2 recessive alleles to have that disorder. So these are ones that aren't as common because you have to have 2 recessive alleles. Now, when I say not as common, like I said, people can carry this, but a lot of times some of these diseases are fatal. For example, if a child has Tay-Sachs disease, they die at about the age of 3. And in the past, individuals with cystic fibrosis died fairly early. Most of the modern medicine, though, is allowing people to live fairly normal lives with cystic fibrosis. But there's still problems with diseases like sickle cell anemia, which is also a recessive disorder. So that's why they're not as frequent, is because when you get some of these diseases, they actually kill you before you are able to reproduce and therefore don't have a higher probability of being passed on. But they're still maintained in populations, and we'll explain why in the next lecture in evolution. Because there is an advantage sometimes of carrying these diseases, believe it or not. All right, so autosomal recessive. Now, in this scenario, there's a new word you're going to have to learn. It's called carrier. Carrier is synonymous with heterozygous. Okay? Because a carrier is an individual that has a dominant allele. Well, let's not use H. Let's use T for Tay-Sachs disease. A carrier is an individual that has a dominant allele, and because this disease is recessive, that makes them have a normal phenotype. But they carry the recessive allele that would cause the disease. So their phenotype is normal, but they do carry that recessive allele with them that they could pass on. That's why they're called carriers. Okay? So carriers, this only applies to recessive disorders, not to dominant disorders. Okay? So if you're looking at a dominant disorder, like Huntington's disease, we wouldn't call this individual a carrier because they will have Huntington's disease. They do not have a normal phenotype. So that's why it doesn't apply to dominant disorders. It only applies to recessive disorders. Okay, so carrier means heterozygous. Okay. Now, in order to have the disease, you must have both recessive alleles. So let's do cystic fibrosis. Okay, let's say two parents have a normal phenotype. They don't have cystic fibrosis, but they are both carriers for cystic fibrosis. What are the chances that they'll have a child with cystic fibrosis? So let's look at their alleles. What are the alleles of each parent? They're both carriers, which means what? Heterozygous, and they have a normal phenotype. So what are their two alleles? Big C, little c, because this is normal, that's cystic fibrosis. You have to have the two recessive alleles to have that phenotype of cystic fibrosis. That's the difference between recessive disorders and dominant disorders. Okay, so what's the other parent? Big C, little c, because they're a carrier. All right, so big C here, little c here, big C here, little c there. What are the chances they'll have a child with cystic fibrosis? 25%. Okay? That's where that statistic comes into play. All right, let's do another one. Let's say one parent has cystic fibrosis, and the other parent is a carrier of cystic fibrosis. Okay? So the parent that has cystic fibrosis, what's their genotype? Little c, and what about the parent who's a carrier? Okay. What are the chances that now that they'll have a child with cystic fibrosis? 50%. Okay. What if one parent has cystic fibrosis and the other parent doesn't, and that parent is not a carrier? What are the chances they'll have a child with cystic fibrosis? 0% chance that they'll have a child with cystic fibrosis. So if one parent has cystic fibrosis, the other parent doesn't and is not a carrier, meaning they're homozygous dominant, because that's what carrier means, then there's no chance for them to have children with cystic fibrosis. But it's 100% chance that all their children will have the allele, will have the recessive allele. Sex-linked is different because now we're dealing with non-homologous chromosomes for the males. Men, you get the short end of the stick when it comes to diseases on the sex chromosomes. Because instead of having two alleles, we only have one. Now, when we look at sex-linked inheritance, we don't look at the Y chromosome. Why? Number one, there's only 100 genes on here, and mutations in this gene typically cause sterility. So they don't get passed on from one generation to the next. So we really don't look at the inheritance of mutations on the Y chromosome, because any mutations here cause major problems. But the X chromosome has over 1,000 genes on it, and some of them have nothing to do with the sex development of the male and female, so they don't cause sterility. One or two examples, and these are the two that I'll be using for your quiz and whatnot. Color blindness and hemophilia. These are sex-linked. This is why men are more prone to color blindness than females are. So let me explain how this works. Now, color blindness and hemophilia are recessive disorders. So we're only going to deal with recessive sex-linked inheritance. Yes, there are dominant alleles, but for my class, I'm just going to deal with the recessive for the sake of it. Okay? So why do men get more affected by recessive alleles than women do? Well, here's the thing. Women have 2 alleles for every gene on the X chromosome because their chromosomes are homologous. They have homologous chromosomes. Men, on the other hand, we only have 1 allele for all of our X chromosome genes, which means that we either inherit the dominant allele or the recessive allele, but not both. We don't have 2 alleles. We've got 1. Women, on the other hand, have 2 alleles. So it follows the same rules as the other recessive disorders. Color blindness being a recessive disorder, women have to actually inherit 2 recessive alleles to be colorblind. Men, you get that 1 recessive allele, there's no other allele on the Y chromosome to mask that you're colorblind. So that's the nature of sex-linked inheritance. Hemophilia is the same thing. Men are more prone to it because it's on the X chromosome. 1 recessive allele, and you've got it. So let's look at a scenario. Now, people tend to get confused on the sex-linked inheritance for one main reason. They forget men only have 1 allele. So here's a trick to make sure that you don't make that mistake. Draw the sex chromosomes first. When you do that, then you remember, oh yeah, there's no allele on the Y chromosome. Okay? So, when doing the Punnett square, you'll still look at the probabilities, but you'll see that it shows the outcome for the males and it shows the outcome for the females, because now it matters what the sex of the individual is and the statistics that arise from that. So what I typically do is if a woman is colorblind, then I put the little C right next to the X. If she's not colorblind but she's a carrier, then it'll look like this. And if she's not colorblind and she's not a carrier, then it'll look like that. Okay? Men, we have an X and a Y, so we either have the dominant allele and we're not colorblind, or we have the recessive allele and we are colorblind. Okay? So let's look at the first scenario. Let's say a mother is colorblind and the father's not colorblind. Now here's the thing that also people get tripped up on. All I have to say is the father's not colorblind. And a lot of times people would be like, well, I don't know whether he's homozygous dominant or heterozygous. He's neither, because that doesn't apply anymore to the sex link. We either have the dominant allele or the recessive allele. So if men are not colorblind, what's their allele? It's the big C, it's the dominant. If women are colorblind, since this is a recessive disorder, what are her alleles? Little c, little c. All right, let's put it together. You might want to make your Punnett squares a little bigger too. What are the probabilities that the daughters will be colorblind? 0, because though they are all carriers, they can't be colorblind because they need 2 recessive alleles. What are the chances the sons will be colorblind? 100%. 100% chance that the sons will be colorblind. So if your mother is colorblind, then the males are going to be colorblind. That's just the nature of it. Because it doesn't matter which X chromosome she passes on, you're going to get the recessive allele that causes color blindness, and there is no other allele to mask that. There's no dominant allele or any other allele to do that. That's why males are more susceptible to a lot of these recessive sex-linked disorders than females are. You have a question?
Sorry, my mom is colorblind, and
Yes.
my brother got it as
Yeah.
well.
Mhm.
So would that make my grandpa colorblind?
In order for a woman to be colorblind, the father has to be colorblind, yes. And the mother of of your mother, so to speak, would at least have to be a carrier for that to occur. Now, so both parents don't have to be colorblind, but the father has to be colorblind, and the mother at least has to be a carrier, and then that combines and causes colorblindness in women. So yeah. So once a woman is colorblind, all of her sons will be colorblind. That's just the nature of it. All right. Now, those are 3 out of your 7 Punnett square questions that you're going to have. So there's still quite a bit more to go through as we go through here. We're not going to do any pedigree charts, but this just illustrates some of the dynamics of inheritance. For example, if you're looking at a chondroplasia, which is a dominant disorder, individuals which have a chondroplasia, it doesn't skip generations. if it doesn't get passed on, then it's not going to continue to be passed on from one generation to the next. But if it gets passed on, then it, because it works in a dominant fashion, then it can continue to be passed on. Recessive traits, on the other hand, recessive disorders like albinism in this case, can actually pop up where you're like, wait, this generation and this generation didn't have it. Now, the half means carrier, but they don't actually express the recessive disorder, in this case albinism. So here is where you can see how individuals which are both carriers, they can have offspring where, because of the recombination, it's a 1 in 4 chance, but that's still pretty likely, that they inherit the 2 recessive alleles and therefore have albinism. So this is where you can have a couple generations where you don't see the disease, and then all of a sudden it pops up because it's a recessive disorder. Here is an example of a sex link, the color blindness, as I mentioned, where neither parent are colorblind, but here the woman is carrier. So when she has a son, she really has a 50-50 chance of whether or not she passes on the X chromosome with the normal allele or the X chromosome with the recessive colorblind allele. And in this case, this son's normal, but in this case, the son inherited the recessive allele and therefore is colorblind. Over here, they had a daughter, and she happened to inherit the recessive allele from her mother, and then she got the normal allele from her father. But then when she ended up having 2 sons, both of them inherited the X chromosome with that recessive allele, and therefore they're colorblind. Whereas here, if you have a father who's colorblind, it's guaranteed that the daughters are at least going to be carriers. If the mother is not a carrier and she's not colorblind, they're at least going to inherit that, because if they're a girl, then the father had to have passed on the X chromosome. All right. Now, let's look at scenarios beyond just the dominant recessive that we've been talking about. Because in reality, a lot of things don't fall under that category where the alleles are either dominant or recessive. A lot do. A lot of the alleles that create your traits are either dominant or recessive, but there are other scenarios where they're not. So this is what we call beyond Mendel's laws, because they look for patterns of inheritance other than the dominant recessive relationship that Mendel was looking at. So here's an example. I'll give you an example of flowers and also give you an example in the human species of this scenario, and either one of those are fair game as far as a Punnett square question for your quiz. So it's either going to be on the flowers or it's going to be on the human scenario I give you. So sometimes alleles aren't one dominant, one recessive. In some scenarios, they're both dominant. But when you have an individual who is heterozygous, meaning they have one of each of the dominant alleles, you end up getting a mixture of the phenotype. And this is what we call incomplete dominance. So here, the alleles either encode a pigment in which makes the flowers red, or there's a different allele that encodes a pigment that makes the flowers white. Now, when you get a cross between these individuals, and you get one red allele and one white allele, it's like mixing 2 colors on a palette. You get an intermediate phenotype. Make sure you understand that word, intermediate phenotype, which is essentially a combination of the 2 phenotypes, and you get something in the middle. All right? So here's a scenario where if you take 2 heterozygous individuals and cross them together, you can see that you ultimately get these statistics of homozygous for the red allele, heterozygous, one red, one white allele, and homozygous for the white allele. Notice again, you don't have dominant recessive in this case. They're both dominant, but we call them incomplete dominance. Neither one is dominant over the other, and so you get a mixing of the 2 phenotypes. What's another example of this? Well, in humans, your hairs, as they grow out of your head, either are round and therefore make straight hair. That's one allele. The other is where the hairs are actually flat and they end up curling up on themselves, and that's what makes curly hair. Now, for the sake of making sure you understand these are the same gene, we're going to do S1 and then S2. Okay, so these are dominant alleles. This one makes straight hair. This is curly hair. What do you think the intermediate phenotype is? Wavy. So naturally wavy hair is an example of incomplete dominance, where you end up having one of each allele and you get this intermediate phenotype. This is incomplete dominance. Okay? So that's really another example in human genetics.
Does this exist for recessive genes too?
I can't think of any examples. I can't think of any. I'm sure there are, but I can't think of any off the top of my head. Let's see. So, let me give you a scenario. Let's say that you had two parents with naturally wavy hair, where they have this intermediate phenotype. What are the chances that they'll have a child with curly hair? So, straight hair, you have to have two of the straight hair alleles. Curly hair, you have to have two of the curly hair alleles. So I'll do this. Okay. And then you have two parents who have wavy hair. What are their genotypes? S1, S2, and S1, S2. Okay. So, we'll do the Punnett square, S1, 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 S2, S1, S2, and S2, S2. What are the chances that they'll have a child with curly hair? It's the same as this right here. Okay? This is in fact the same scenario. If you were to cross two pink flowers with each other, you get 25% chance that they're red, 25 or 50% chance that they're pink, and 25% chance that they're white. In the case of these two parents, 25% chance that their children have straight hair, 50% chance that they have wavy hair, and 25% chance that they'll have curly hair. Okay? So it's going to be one or the other of those. We call these 4 o'clock flowers. There's a number of different types of flowers. There's also snapdragons, which I think is what these are as well. But there's several types of flowers that show this same type of relationship with each other. All right. Now, the last few Punnett square questions you're going to have are on this last concept. So you've got a couple on blood type. But this is one of those that people find pretty fascinating because they're always interested in understanding how blood type works. All right. Now, before we can get into the Punnett squares of this, we first have to describe a little bit about how blood types work and what they are. Okay? Now, if you remember back from cell biology, we talked about what was called a glycoprotein, which is essentially a protein with a carbohydrate attached to it that's embedded in the cell membrane. Now, we use these for things like cell recognition. Remember, these are the oligosaccharides that ultimately determine what is self and what is foreign. So we learned this back in when we discussed cell biology, and we discussed how proteins were made and how the Golgi apparatus attaches protein or carbohydrates to the proteins. Well, that's what we're looking at right here, because that's what your blood type is. It's a very particular glycoprotein that is attached to the surface of your red blood cells. So if you have blood type A, you have what we call the A antigen. So antigen and glycoprotein are the same. Okay, in this case, antigen is that surface recognition molecule that your body uses to recognize what is self and what is foreign. Okay? So some people have the A antigen on the surface of the red blood cells. Other people have the B antigen on the surface of the red blood cells. Some people have neither one, and that's what gives us blood type O. But in some cases, people have both A and B antigens. In fact, this is one of the least common blood types in the U.S., statistically speaking. But there are individuals which are blood type AB. Now, here's why it's beyond Mendel's genetics. Up until this point, we've talked about genes having pretty much two alleles, a dominant and recessive allele, two dominant alleles. Now we're looking at 3 alleles. Blood type is a single gene, we call it the ABO gene, so you can guess why, that has 3 different alleles to it. Now, 2 of the alleles are dominant, and 1 allele is recessive, which is why we use the I as a capital to indicate the dominant allele, and a lowercase I to represent the recessive allele. So if you have the recessive allele, this doesn't make either antigen. Essentially, it's like putting nothing on the surface. Okay? So that's why this is recessive. Because if you have even one allele that makes the A glycoprotein and the other is the recessive, you'll still make the A glycoprotein, you'll still be blood type A. Same thing for B. If you make the B antigen, but your other allele is recessive, you'll still make the B antigen and your red blood cells will be blood type B. If you have both the A and the B antigens, then both of those are expressed on the surface of your red blood cells and your blood type AB. And only if you have the two recessive alleles do you have blood type O. Because in the presence of any of these other two dominant alleles, you'll either have blood type A or blood type B. So blood type O is recessive. Now remember, we talked about recessive does not mean that it is the least common. In fact, 45% of the US has blood type O. So it's one of the most common blood types in the US, AB being the least. The next most common is blood type A. Third most common is blood type B. Now, you'll notice I'm ignoring the RH factor, which is the plus or the minus. I'll talk a little bit more about that later, but for now, we're just going to focus on the A or the B antigen. Now, there are more than just these four genotypes, because in fact, if you have two A antigens, you're also blood type A. And if you have two B antigens, you're also blood type B. So in fact, if you're blood type A, there are two possible genotypes for that phenotype. And if you're blood type B, there are two possible genotypes for that phenotype. It's only blood type AB and blood type O that you end up having one genotype associated with that phenotype. There's only one way to get blood type AB, and there's only one way to get blood type O. So here's where things get a little interesting. Now, here is the concept regarding these. We already know the dominant recessive relationship. That's old news. You already know that. But what happens when you get two dominant alleles? Well, this is not like the example we just went over. This is not incomplete dominance. It's what we call co-dominance. Now, there's a big difference between the two. Let me explain. Co-dominance means that neither allele is dominant over each other. They're both expressed independently. Okay? Neither is dominant. So you don't get this intermediate phenotype. Let me give it to you in a scenario like this. If, this is a big if these flowers' alleles represented co-dominance, what would you expect this flower to look like? Or this plant? Good. Red and white. You would not get anything in between. You'd have some petals which are red and some petals which are white. If we saw the same scenario with the human, the straight and the curly hair, if you plucked out a hair, it'd be either straight or curly, but not in between, not wavy, if it were co-dominant. So really, that's the big difference. Incomplete dominance, you get a mixing of the phenotypes and you get that intermediate phenotype. Co-dominance, on the other hand, you get both of them. You get both expressed at the same time. You get blood type A and B antigens expressed on the surface of your red blood cells, and therefore your blood type AB. Now, let's look at some of the Punnett square scenarios. Now, if I tell you that someone's blood type AB or blood type O, I do not have to tell you anything more. You know what their genotype is. As I mentioned, there's only one genotype associated with blood type AB. There's only one genotype associated with blood type O. It's blood type A and B that become a little tricky because there are two possible genotypes. You could be homozygous for the A antigen, or you could be heterozygous. You could be homozygous for the B antigen, or you could be heterozygous. Now, when we say blood type A heterozygous, you already know that the other one has to be the little I, because it can't be this, because then you can't be blood type A. So if I say, oh, they're blood type A and they're homozygous, the alleles are the same. If they're blood type A heterozygous, then they have one A antigen and then the recessive one. Okay? Because this is the only other heterozygous scenario, in that situation, you can't be AB. So same thing for B. If you're homozygous, blood type B, or heterozygous, blood type B, those are the differences between those genotypes. Okay, now with that knowledge, let's look at some scenarios. If you have two parents with blood type O, What are the chances of a child with blood type O? 100%. This is why they don't genotype or blood type kids in the schools anymore. Because there have been kids that are like, I'm blood type A and my parents are blood type O. Yeah. So ultimately, we don't do that anymore. All right, let's look at a parent who's blood type AB and a parent who's blood type O. What are the chances they'll have a child with blood type A? 50%. Okay. Let's do one parent is blood type A and is heterozygous. The other parent is blood type B and is heterozygous. What are the alleles then? If they're blood type A and heterozygous, what are they? IA, little I, and blood type B heterozygous? IB, little I. And guess what you get? Everything. 25% chance for all 4 blood types. Okay? That would be an interesting family. So, 25% chance blood type A, blood type B, blood type A, and blood type O. Okay? Yeah?
So what is the deciding factor for like a person to get a particular one? So if they had 25% of each one, what is the actual deciding factor of what blood type they get? Or is it just random? Then we just don't.
Well, it all depends upon what in genetics they inherit. So if the father passes on his B antigen and the mother passes on her O antigen, then the child will be blood type B. So it all depends upon when you undergo meiosis and you split your homologous chromosomes, which allele you pass on, and then the combination will ultimately determine that. But here's where you're going to have 4 different possible phenotypes from that scenario. In fact, that's shown right here in this scenario right here. Okay. Now, you're going to have a question like this, but here's the more difficult of the questions. So let's do this. Let's say you have a child who is blood type O, and you have a parent who's blood type A, and another parent who's blood type O. What is the genotype of the parents?
IA, little I.
How do you know that?
Because in order for the O to happen, it has to have recessive.
Good. Law of segregation. In order for this child to be blood type O, we know this parent is little I, but that means that this parent has to be heterozygous. Okay? Otherwise, they can't inherit that second O allele. All right? Let me give you another scenario. Same thing. Parents are A and O. Child is now A. Which of those genotypes can you figure out? Do you know the child's?
Not 100%. Has to be IA.
Why does it have to be that?
It's the only way to make it.
That's the only way. I mean, this person is blood type O, so they only have 2 little I alleles. And because this person's A, they've got to at least have the A antigen. Now, do you know this parent? No. You cannot, from just this information, you cannot figure out because the parent could be this, or they could be this. Either one works. So in that scenario, you would not be able to tell us the genotype of that parent. But you would be able to of the child and of that parent. Okay? Now, before I go on to the last little bit, sometimes people are interested to know why certain blood types can't be mixed with others. So let me just, this isn't any test questions, but just for your sake, for your knowledge. If you're blood type A, then you have the A antigen. We've learned that already. But your body will produce antibodies against the other antigen that is created, which is why we create these anti-B antibodies. Now, the same thing is true for someone with blood type B. They produce B antigens on the surface of their red blood cells, but they'll produce the A antibodies as a result because their body will not needs to recognize anything else that's foreign. Now, there are many other antibodies that are produced besides these, but this is amongst those that are produced by those individuals. If you're blood type A and B, then the body essentially doesn't produce either of these antibodies because this is what happens when you mix the antigen with antibodies. Let's say someone with blood type B, or someone with blood type A gets a transfusion of blood, but it was the wrong blood. Somebody screwed up. They put blood type B and mislabeled it. This is what happens when the blood type gets into the person who has blood type A. The person with blood type A produces these B antibodies. They attach to it. They cause this agglutination response and then the bursting of the cells. It would be the equivalent of pumping mass amounts of bacteria into somebody's body. It will kill them. This immune response, if you pump enough of this foreign blood into someone, will kill them essentially.
What's the purpose of blood cells being able to detect foreign blood?
Well, ultimately, we don't just produce antibodies against the other one. We produce antibodies against everything else. So if you're blood type A, you have millions of different antibodies. Your body will only eliminate ones that will recognize self. So your body has the potential to create the antigens against A, but it destroys those cells that would actually make that. So this is kind of misleading in saying that we only produce that. We produce millions against any foreign antigen. This is one amongst those millions. And if somebody has this blood type, they essentially destroy any cells that would make anti-A and anti-B antibodies of the millions that we produce. So really it's just about recognizing anything foreign. But this is amongst those antibodies that we make. Now, here's the thing here. If you're blood type O, you do not have any antigens, and therefore the body doesn't destroy any of these antibodies against A and B. So here's the thing. Blood type O are called the universal donors. Why? Because when you usually filter out everything but the red blood cells, that's why when you do a transfusion, you're only passing on the cells. There's nothing here to be recognized as foreign. So each of these cell types can receive blood type O without any problems. Now I'm ignoring the Rh factor. We'll talk about that in a sec. If you're blood type A, sorry, but they don't need your blood because they may. There are some scenarios where you might need an AB negative because of some individuals or whatnot, but most of the time they're not in high demand. If you're blood type O, you're in high demand because it's the universal donor. Now, if you're blood type AB, you're better on the receiving end because you can pretty much receive blood from anybody because it doesn't matter whether the blood has A antigens, B antigens, or no antigens. You're not going to reject any of it because of your immune system. Okay? Now, the same thing applies to what we call the plus or the minus. So you say, oh, I'm A positive. Well, those are two different antigens. One's the ABO gene. The other is what we call the Rh factor. And you either have it or you don't. So if you have it, that's where the positive comes in. If you don't have it, that's where the negative comes in. Now, this applies to actually pregnancies. If women give birth to a child who is Rh positive and they themselves are Rh negative, the first child won't have any problem. But in the birthing process, there's usually some mixing of blood, and when the mother's exposed to that Rh antigen, then she'll start producing antibodies in surplus against that. The next time she has a kid, then those antibodies can actually attack the fetus as it's developing, and these mothers usually have to go on immunosuppressive drugs to be able to have the child because of that issue. Now, it doesn't, for whatever reason, it's not an issue if the mother's blood type O and the child's blood type A. That's not the problem. But that Rh factor somehow crosses that placental barrier, and the antibodies can actually attack the fetus's blood. So, anyway, that's blood type in a nutshell. Now, let's talk about the last set of relationship between genes, alleles, and traits. Everything up to this point, including the exceptions, the blood type, which has 3 alleles, the incomplete dominance, like with the straight and curly hair, those are all what we call Mendelian genetics. And the reason why we call them Mendelian genetics is because they're one-to-one relationships. One gene, one trait. You know, you've got the ABO gene, which, yes, makes multiple types of blood types, but it's still one gene, one trait. Most things in most organisms are never that simple. Most of the time, our traits are what we call polygenic. Poly means many. Genic means genes. So in reality, most of our phenotypes, even something that you might consider as simple as hair or eye color, are polygenic. That means that it actually requires multiple genes and their respective alleles combined together to create your hair color, your eye color. So it's not just a one-to-one relationship. So polygenic, this is how most things are. Even your skin. Your skin is determined by as many as 6 different genes with their respective alleles. In fact, this is kind of an example. This only shows 3 different genes labeled A, B, and C, but in reality it's more like 6 different genes. So this just kind of illustrates that you have dominant recessive alleles for each gene, and depending upon how many dominant and how many recessive alleles you have for those, ultimately you determine your natural skin pigmentation. So, polygenic, many genes, 1 trait. This is for hair, eye color, height, you name it, most of the time this is what it is. This is why it's not always as clear-cut and simple to say, oh, both parents have brown hair, then you're going to have a brown-eyed or brown-haired child. It's not that simple. It's a little more complex than that. Okay. Now the next concept, there are scenarios where the opposite is true, where one gene can actually influence multiple traits. We call this pleiotropy. Now, we've talked a little bit about Marfan syndrome. This is actually a dominant disorder, a dominant gene. However, there's not just one symptom of Marfan syndrome. Some people have skeletogenic abnormalities. Other people have heart problems. Other people have lung problems. Other people have skin problems. Why? It's a single gene. It's a connective tissue gene. It has to do with creating the fibronectin and whatnot in the connective tissues. But all of these have connective tissue in them, and depending upon what happens during development, you can have any number of phenotypes associated with this one genetic disorder. In fact, that's what they found with some family trees, is they were looking at genetic disorders. Here's one where it's a porphyrin mutation. And they found that some people had mad behavior, others had constipation, dark-colored urine. From generation to generation, there were different symptoms. And they're like, man, you guys are all screwed up. Well, it's the inheritance of one gene, one problematic gene from one generation to the next. So that's pleiotropy. A massively severe case of pleiotropy that's interesting in of itself. It's not inherited. So it stops after the mutation occurs, but it's called androgen insensitivity. So what is this? It's a female that is XY. So how does that happen? Well, remember I told you when you get defects on the Y chromosome that they're usually not inherited? Well, this is a mutation in a protein, a receptor that responds to testosterone. In women, because they are women, that have androgen insensitivity, they actually have testes instead of ovaries. Now they sit where the ovaries should have been, but they develop testes. But due to the mutation in this one receptor on the Y chromosome, the testes secrete testosterone, the body can't respond to it, so it develops as a female. So it has all of the secondary characteristics of a female, from breast development to long hair and everything. The only difference is they don't have a uterus. So women, if you've had your period, you don't have androgen insensitivity. Okay? So you're not XY. But this is like a 1 in a billion chance for this to happen. It's not very likely. And it's not inherited, because obviously it causes sterility as well. So that's just another extreme example of pleiotropy, where one gene can actually change the entire sex of the individual. Instead of being male, as female. All right. Now, 2 more concepts. Sometimes we see scenarios where 2 people have the same phenotype, but all of their children are normal. And so the question becomes, well, how does that happen? In fact, 2 different examples. You've got dwarfism and you've got deafness. And these are genetics that can be inherited. But I grew up with friends whose parents were deaf, but all of their children were hearing. And then there are times where you have 2 parents with dwarfism, but all their children are normal height. So how does that happen? Because statistically, you would at least expect 1 or 2 of them to have the same genetic disorders. Well, this is what we call genetic heterogeneity. Basically, you arrive at the same phenotype, but you have different mutations in different genes. And the reason for this kind of goes back to polygenic inheritance. multiple genes all combine together to form a particular phenotype. Well, if you mutate one gene, then you cause some problem in that ultimate phenotype. If you mutate a different gene, you may cause the same ultimate result. So that's why two parents with dwarfism may have dwarfism in common, but not because of the same genetic abnormality. And when they have children, because they have different genetics, the recombination of those genetics restores normal functionality, and all their children to normal height, or all their children can hear. Because there's many genes that go into creating the hearing of the cochlea and the inner auditory ossicles, and basically hearing in general. Blood clotting as well is another example of this. There are many different genes all coming down to the process of blood clotting. So if two people have poor blood clotting, it may not be for the same reason. So that's genetic heterogeneity. Now, can two parents with blood type O have a child with blood type A? Yes, but it is rare. But it illustrates the last concept called epistasis, where one gene can actually supersede the expression of another. Now, we've already gone over one example of this. Let's go back. I didn't point it out at first because I didn't want to add more on, but let's go back. Albinism is an example of epistasis. So what happens in albinism is, we talked about how there are about 6 different genes that cause the skin pigment colors to occur. But there's one gene to control them all. Yes, Lord of the Rings scenario. One gene that ultimately, if you mutate that gene, it sets everything at a default and goes down to what we call albinism, where the hair is light colored, the eyes are light colored, the skin is completely white, there's no pigmentation whatsoever. And that's what you're seeing here in this family, is that these kids should have darker skin, but that one gene supersedes all of the others in its expression and ultimately makes it so that they don't have any skin pigmentation whatsoever. Now, what's another scenario? I talked about blood type. Let me show you how this works. We know that the ABO gene makes the antigen, and depending upon which antigen gets attached to the surface of the red blood cell will determine what your phenotype is. Okay. Well, guess what? There's a gene that supersedes this one that ultimately attaches the antigen to the surface. If you have a mutated version of this gene, it doesn't matter what your genotype for the ABO gene is, your default is going to be blood type O. Even though you make the A antigens, it never gets attached to the surface of the cell because of the mutation in this gene. So in this scenario, the person's phenotype is O, but their genotype is this. Now, take another person who is actually blood type O because of the genotype, cross them together, what's the child's genotype going to be? Or phenotype's going to be? It's going to be A. Now, how does this occur? How does it, when you recombine these, all of a sudden the normal functionality is restored? Well, in this parent, this gene is mutated, so it doesn't attach the antigen to the surface. Over here, they have normal genes. When you recombine these, it restores the functionality of that gene, and now this can be expressed just fine. This is what we call the Bombay phenotype, where two parents with blood type O can have a child with a different blood type, with blood type A or whatnot, but it's very rare. So it's not one of the things you would look for first in that scenario. Okay, now, there's not a question on this, but this is another interesting concept that I want you to keep in mind, and then we'll do one last question or scenario to finish off this lecture. Most of the time people ask the question, are we genetic, are we nature, or are we nurture? We're both, okay? We're both genetic and environment, okay? Sometimes environment can have more of an influence over our phenotype than the genetics do. What's an example of this? Your fingerprint. Your actual fingerprint, not your DNA fingerprint. Your actual fingerprint is about 90% environmentally influenced, 10% genetically influenced. That's one huge scenario. Now, on the other flip side, as we talked about with blood type, that's pretty much 100% genetics with no environment. So it all depends upon what trait you're looking at to determine what the overall relationship between how much genetic influence, how much environmental influence. Now, to give you a scenario of how environment can really kind of screw things up, let's look at this Siamese cat or the Himalayan rabbit. What do you notice about both of them? They're black in their extremities, but their core fur is white. Well, here's what they found. They don't have multiple genes going on here. They just have one gene that's defective. And when the temperature in their core body gets too high, then it doesn't make the black pigment. That's why the extremities are black, but the core body, where most of the heat is held, is white. Now, how do they know this? Well, like scientists do, they strap an ice pack to the back of the Himalayan rabbit and let the fur grow out. And then when they pull it off, boom, it's black. So they showed by lowering the surface temperature of their skin that that's exactly what this is supposed to do. But because of the environment or the temperature of the rabbit, then it defaults to white because it messes up the protein. Anyway, an interesting point. But to illustrate that we're both nature and nurture. We're not pure genetics, which is why if you clone yourself, you're not going to have the same individual unless you have the exact same environmental influence in both in fetus, in embryo, as well as throughout your life. Otherwise, it's no different than identical twins that start out genetically the same, but due to environmental differences ultimately become different over time.